Hi, everyone, and thank you for attending CIL's Isotope Day, featuring science facilitated by stable isotopes. Isotope Day has been divided into two parts, with this session devoted to MS applications and a second one on October 4th devoted to NMR. I am Andrew Percy. I'm the MS Omics Product Manager and a Senior Application Scientist at CIL, and I will be your host for today's event. This webinar features presentations from 6 prominent scientists in the MS field. Their target works are diverse, so the presentation content should provide you with good insights in how to approach different research problems with the aid of stable isotope-labeled standards. Through today's webinar, I hope that you learned something new, strengthen your understanding of CIL's labeled products, and most importantly, stay engaged and have some fun. The agenda for today's webinar is outlined here. Following this brief introduction will be 7 presentations that will cover a broad scope of topics as outlined in this table. Each approximately 15-minute talk will be directly followed by a Q&A period. Please type your questions into the Q&A box at any time throughout the session, and I will direct these to the appropriate speaker as time permits. Any lingering questions will be answered by the speaker during the event or via email afterwards. Before moving into the scientific talks, I'd like to take a brief pause to ask that you all complete this brief survey. This survey will help us gain a sense of the audience background, and it consists of 3 multiple choice questions. And it should not take longer than half a minute. So I will pause and allow you guys to complete that. I give another 10 or so seconds. All right, we could carry on. Christy can close the poll. Now move into the scientific talks, beginning with my opener on guidelines, considerations, and benefits of incorporating QCs into MS Omics experimental practices. The omics of my talk will center on is metabolomics and proteomics. Examples of pre-formulated multi-component mixes that are amenable to QC measures in these omics disciplines will be discussed. Guidance on their implementation will be provided, and results of their use will be discussed. First, some brief background. Methods in metabolomics and proteomics in particular are multi-step, which can impose degrees of variability that can ultimately impact the accuracy and reproducibility of the acquired results. To monitor and correct, QC measures must be implemented. These can take different forms and be utilized in different ways, each with or without stable isotope-labeled standards. Highlighted here are some articles that discuss these QCs in varying depth. Also important in the realm of QC is a QC of the standards and mixtures themselves. This must be conducted in accordance with SOPs and ultimately return consistent results from vial to vial and lot to lot. This is important such that the QC material itself is not in question over the course of its analysis. CIL offers a broad and diverse portfolio of stable isotope-labeled and unlabeled compounds. The total exceeds 15,000, with their uses covering a broad spanning array of research areas, a few examples of which are displayed here. Today's speakers will cover product use cases from several of these research buckets. For QC purposes, a collection of individual standards could be selected, then self-formulated into mixes, or our already formulated multi-component mixes could be selected for direct use off the shelf. Our multi-component mixes are prepared to exacting standards following detailed batch records, with the inputs and outputs all qualified using carefully designed quality control tests. The mixes are primarily neat, with the analytes being at varied isotopic compositions, having defined concentrations, and the mixes having established specification guidelines in terms of storage and stability. The benefits to using our pre-formulated mixes are numerous. Helps reduce development time and cost, provides flexibility in use, enhances data quality, among other benefits. We offer an extensive and expansive number of metabolite-based and peptide-based mixes and kits. These can be mixed and matched according to your experimental preferences, then applied for use in quantification and/or quality control exercises. Please refer to our mixes and kits catalog and listing shown here for a more comprehensive product overview. There are a few mixes that I've listed here that I will discuss today in the light of QC. Now we'll begin with the Metabolomics QReSS mixes. QReSS is supplied as individual mixes in its labeled or unlabeled form, as well as in a kit, in which case the mix is additionally accompanied by a document package. That package is accessible via QR code. The compositions of the labeled QReSS mixes are tabulated here. The metabolites span a breadth of metabolic classes, as shown in the pie chart, possess a spread of physicochemical properties, and are concentration balanced to endogenous levels found in normal human plasma, as verified against one type of metabolomics method. Development of the QReSS mixes was done in collaboration with SCIEX. This involved a series of characterization tests within and between batch lots in the absence and presence of matrix, all while measured on a plethora of high-res and low-res instrument platforms. These chromatograms illustrate representative RPLC separation of the QReSS metabolites measured under positive and negative ESI. Overall, from the QCs, the entire 18 target QReSS panel was measured reproducibly with stable metrics obtained across vials, replicates, and lots. Please see our joint application note number 49 for an overview of method details and the results. Using those qualified QReSS mixes, we have embarked on a number of collaborations with MS vendors such as Thermo and academic groups. A recent example of one of those academic collaborations is with the Lorenzi Lab at MD Anderson. Here, the QReSS mixes was aimed to be incorporated into an analytical pipeline for both daily and in-application performance testing. These experiments used diluted solutions of the combined QReSS mixes, then for use in both neat and pooled in individual matrix samples. Measurements were all made by Helic MS under positive and negative ESI. Data analysis was performed in Trace Finder as well as Excel. Briefly on some results. System suitability testing involved measuring the combined labeled QReSS mixes neat on a daily basis with Helic and MS characteristics tracked over time. The monitored features generally return stable outputs, but there were some cases where notable deviations were observed. Highlighted here in this figure, as shown by the boxed out data, is a point in which an error was observed. And this relates to retention times that was attributed to an air bubble in a mobile phase line. A system purge corrected that issue, restoring performance back to baseline levels. Given the importance of having accurate and reproducible RTs in compound ID and quant experiments, this data exemplifies one of the benefits of running system suitability QCs as part of daily instrument operation. For in-application performance testing, a batch sequence was designed with a number of QCs interspersed around individual matrix samples. In this case, the matrix was cancer patient plasma samples, and there were 41 of them. The QCs included extraction blanks and system conditioning blanks, while the labeled QReSS mixes was used in both a neat sample as well as in a pooled matrix sample, as well as being spiked into the matrix for quantitative purposes. Overall, from the QCs, we found negligible QReSS metabolite signals in the blanks and stable and precise metrics observed across the QCs. In this chart, in this figure, it shows labeled QReSS metabolites for both the pooled QC and across all 41 plasma samples. And that indicates the stability for a subset of QReSS metabolites. In development right now is the tolerance criteria for the QReSS metabolites across different types of QCs, as well as a software tool for automated real-time metric tracking. Now, QC mixes need not be restricted to those that are designated or designed for that purpose. Our bile acid mixes, for example, is one of those cases. These bile acid mixes comprise 6 unconjugated BAs in one vial and 10 conjugated BAs in a second in their partially deuterated or unlabeled form, all at equimolar concentrations. In a bile acid study to investigate liver disease in mouse models, the labeled bile acid mixes were used not only as IS in quantitation, but they were also used in a number of different QCs. The high quality of the QC results demonstrated that the workflow was indeed fit for purpose and provided confidence that the application data was acquired effectively. Dr. Amy Engevic will present the biological implications of this application work later in her talk of this webinar, while the joint application number 52 shown here can be sourced for methods, insights, and findings. Switching gears now very briefly to proteomics, the general framework for a bottom-up proteomics experiment can be quite extensive, which can open up many opportunities for error. To help with QC, MRM Proteomics has developed CPTAC certified assays for both day-to-day instrument performance evaluation, as well as method-based workflow performance evaluation. These kits are called PeptiQuant Plus Assay QC kits and are, as indicated, supplied in the daily and workflow flavors. These kits are comprised of a series of lyophilized reagents and a USB of files. Illustrated here in this bar chart is an example of response data for one peptide from the daily QC kit panel. As indicated by the arrow, an error in the labeled peptide response was observed, and instrument maintenance, so, restored performance back to baseline levels, as evidenced by the next collection point in this data series. And that helped pave the way towards the next set of data collections, which are biomarker-driven application collections. So this further reinforces the need for system suitability QCs as part of day-to-day QC operation. So briefly in conclusion, QCs are important and should be run routinely, both independently and in conjunction with experimental designs. A few examples were shown here in my presentation of the benefits of incorporating them from both a metabolomics and a proteomics perspective. Efforts continue with our collaborators to incorporate these into analytical pipelines, as well as develop novel software tools for automated real-time metric tracking. Acknowledgements are extensive, but for the work presented here today, the PIs and scientists I've listed should be recognized. So that brings me to the conclusion of my presentation. I think there might be time for a question or two if there are any. Otherwise, I'll push forward to the next talk. It doesn't appear to be, but you can feel free to email me at any time. And I'll be happy to address any questions.